Excitability in the brain can lead to neonatal seizures. Therefore, being able to decrease the neuron excitability could possibly be an effective way to treat seizure episodes. So then how do we decrease excitability? Well, one way to do this is through regulation of channel proteins. Our lab has been studying the effects of the neurotransmitter GABA on calcium channels in the hippocampus, which is a site of the brain commonly associated with seizures. Now, the hippocampus has a large concentration of GABA receptors compared to the other areas of the brain. And it has been shown that activation of GABA B receptors in select neurons leads to an increased amount of calcium ions that flow into the cell through L-type calcium channels in newborns. This pathway does not exist in adults. It is only present during the early developmental stages of the hippocampus neurons in babies. This unique property of GABA found in newborns may provide the key to treating seizures at such a young age. When GABA binds to the receptor and calcium influx enhances, calcium goes on to play several different roles in the cell. One important role of calcium is the activation of certain potassium channels, and potassium channels play a huge role in membrane excitability. When a neuron gets stimulated, it creates an action potential. A stimulus causes the membrane to depolarize, or become more positive, by opening sodium channels. At the peak, sodium channels close and potassium channels open. The open potassium channels repolarize the cell back towards resting membrane potential. However, the potassium channels stay open a little longer than necessary, creating a hyperpolarization. Many different kinds of potassium channels are involved in the action potential. And, as I said before, some of these potassium channels are activated by calcium. My project will be exploring how activating the GABA B receptor, and therefore increasing intracellular calcium, will affect the hyperpolarization of action potentials. In theory, increasing calcium in the cell will activate more potassium channels and create a longer lasting after hyperpolarization. Action potentials would become more spread out as a result. If this is true, it is possible that neonatals with an overstimulated hippocampus, such as what happens during a seizure, could be calmed down by activating GABA B receptors. By further exploring this mechanism, we hope to become one step closer to solving the problem of neonatal seizures.